environment. All right, guys, good morning. I guess the sooner we get started, the sooner we can finish. Happy New Year, everyone. I want to apologize for bailing on your osmosis lecture in December, but I'm sure Dr. Sullivan did a better job than I ever could have. We're going to talk about the endothelium first, and then we'll follow that with smooth muscle contraction. So the endothelium, why are we going to do this? Endothelium tends to get overlooked. People think of it as a single cell layer. It's saran wrap. Why do we need to learn about it? I really want you throughout this lecture to think of the endothelium as an organ, a secretory organ, almost like the liver. Okay? Really, the endothelium is hugely important, and we'll emphasize this in our Block 8 lectures when we talk about vascular biology. So, just a general introduction to the endothelium. You can read this for yourself, but if you spread all the endothelium out in your body, it covers a pretty large size area, about half of a tennis court. The endothelium is a confluent monolayer. It lines the inside of all blood vessels and many other organs as well, too. It must be an adaptable cell type. It has to respond to environmental changes. It's really important. The endothelium separates the circulation from the rest of us. So it has to be aware. It has to know what it's doing. Otherwise, if you do not have a healthy endothelium, you don't have a healthy organism. It's really important. Since it's a confluent monolayer, by necessity, they need to be polarized. So the apical surface is in direct contact with the blood, interacts with everything, interacts with platelets and leukocytes. And in Block 8, we'll talk about the concept of thrombogenicity. You want the endothelium to be slippery. Okay? The basal surface is in contact with extracellular matrix and in several cell types. If it's an artery, we're talking about a smooth muscle cell. If it's the heart, we're talking about myocardium. Endothelial cells are interesting in that they're heterogeneous. There are many different cell types of endothelium. So a coronary artery or endothelial cell is very different than a lung microvascular endothelial cell, which is also different than, say, an umbilical endothelial cell. And if you think about it, they have to be different because they're in different anatomical locations. The type of endothelial cell also is susceptible to different types of diseases based on its location, and we'll talk about that in about 20-25 minutes. So it is a barrier between the circulation and the rest of us. It needs to sense changes in flow. It needs to detect differences in circulating cell-borne particles, platelets, white blood cells, and things like that. So the endothelium, I like to think of as the brains of the vasculature or the brains of the GI tract or of anything that it lines. Okay, so we talked about heterogeneity of the endothelium. We can really group them into three types based on their structure. And these are based on pathological observations at the turn of the last century. Okay, so we have a continuous endothelium. It is represented by an uninterrupted barrier between endothelial cells, really tightly lined. A good example of the place where this occurs is the blood-brain barrier. You don't want really many substances to leave the circulation and go right into your brain. The second type is called fenestrated endothelium, and it features pores between the endothelial cells as well as inside individual endothelial cells, and they're called fenestrae. So think of it as a layer of the plasma membrane, almost like a porthole or a window, and its purpose is to allow substances to leave the circulation and enter you much more easily. They're primarily found in places where water is reabsorbed, such as the kidney. Then we have discontinuous endothelium. It's also referred to as sinusoidal endothelium. And that's characterized by large gaps between endothelial cells. Why do you think that would be? To allow things to leave the circulation or enter the circulation very easily. So the liver is always absorbing materials from the circulation. Place I like to think of it, a good location for discontinuous endothelium is the bone marrow. So think about it. The bone marrow is constantly making white blood cell precursors. They need to be able to exit that bone marrow and enter the circulation rather easily. You don't think that they could squeeze through a continuous or a fenestrated endothelial layer, but when there are large gaps in the endothelium, they can do that. Here's some electron microscopy. One picture is worth 1,000 words. We want to talk about a structure called caveolae. They're not unique to endothelial cells, but endothelial cells are highly enriched in caveolae. That is Latin for little caves. They're really just invaginations of the plasma membrane. And why are they there? Well, when we had our signal transduction lecture, I told you that there tend to be hubs of signaling. Well, in these caveolae are clustered many receptors. So this is really a hub of signaling, but also it allows the endothelial cell to recognize many substances from the circulation, and they will pinch off and transcytose to the other side of the endothelium. And I have a really good slide on that in about 10 minutes or so. There are also pores between endothelial cells. So here's a nice picture. So this is an endothelial cell. This is a second endothelial next to it. And here you can see the fenestrae. These are really, I like to think of them as just plasma membranes, and it allows water and other substances to traverse the endothelial cell layer rather easily. And again, as I showed you in this picture, you can have fenestrae within an endothelial cell. So when you think of the endothelium, think of the real estate rule. Location, location, location. The type of endothelial cell and its anatomy and what it's based on is based on the physiology it needs to perform. So if you think of kidney, you need to reabsorb water. If you think of blood-brain barrier, you need to keep things out. So one other word I want to mention, I should have mentioned this up front. Like my signaling lectures, I show a lot of slides, but just about everything is on the handouts I provided. I have extra slides up here, such as this one. This is an example that aren't in your handouts. It's just to help me illustrate a point. Study from your handouts. Your colleagues that skipped class today, remind them, study from the handouts. So this is an example of what I'm talking about with the endothelium. It's really prevalent when we talk about capillaries. So we have a continuous capillary, tightly joined together, and where do you think you'd find that? Blood-brain barrier. Fenestrated capillary, you can see all the portholes, all the fenestrae, allows water to be reabsorbed rather easily. That would be in the kidney. And here we have the sinusoidal capillary. That would be in the liver, or bone marrow, and bone marrow. Again, there are large gaps that allows things to be reabsorbed or allows white blood cells to exit the bone marrow easily without tearing the endothelium apart. More EM, a picture's worth 1,000 words. In the capillary, these fenestrated endothelial cells are lined up in arrays, and they're called sieve plates. And this is a really neat picture because you can see how these sieve plates look. When you were a little kid and you played in the sandbox and you had these little screens to play with and allows the sand to fall through, that's what it really looks like. And it allows very efficient uptake of water. So I did my best to sort of categorize the function of the endothelia and endothelial cells into really 5 basic functions for you to hang your hats on. So I mentioned that the endothelia is an organ. They secrete many factors: nitric oxide, clotting factors, vasorelaxation, and vasodilatory factors. Surface expression of proteins. So we'll really talk about this quite a bit in Block 8 when we talk about vascular pathology. But you want the endothelium to be non-thrombogenic. You want them to be slippery. And what regulates that are the expression of molecules on the surface of that endothelium. Contraction. You don't really think of the endothelium as contraction. You think of muscle as contracting. And we'll talk about smooth muscle cell at 11 o'clock. But endothelium can contract and retract away from each other as well. And that allows differences in permeability. Again, they're like the sentinel of what's going on in the circulation versus the rest of you. So they, at certain times, will contract away from each other or tighten up when they feel they have to. And we'll talk about the mechanism of that. They're mechanotransducers. They can sense changes in flow. And angiogenesis. The endothelia can sprout and provide new connections between existing circulatory beds or new blood vessels when they need to. And we'll spend the last part of this lecture talking about that. Okay, so this is a busy slide I took right from the textbook. I only want you to focus on one thing. I showed the entire table because, just to give you an example of that the endothelium can synthesize many different products. It is a secretory organ. Don't memorize this. I have an asterisk next to nitric oxide because we're going to talk about nitric oxide because it's a nice introduction to the next lecture. Nitric oxide is a vasodilator. Why would you want that? So think of an artery. You've had your anatomy. You've got a single cell layer of endothelium lying on the inside of many smooth muscle cells. Smooth muscle cells don't know when to relax or contract and increase the diameter. It's the endothelium that tells them that. So this is important, and I think you should know this. Let's talk about nitric oxide. So the big picture is nitric oxide is made in an endothelial cell, and it's a gas, and it diffuses to a smooth muscle cell. So that's paracrine signal transduction, cells that are next to each other. It's a gas generated through precursors to cause smooth muscle cells to relax. It's regulated by two different enzymes. eNOS, endothelial nitric oxide synthase. It's constitutively expressed. It's an enzyme that's always on. Then there's iNOS, which stands for inducible nitric oxide synthase. That's activated by cytokines and injury. So which one of these do you think a pharmaceutical company would want to direct their drug toward? The iNOS, right? Because you need it when you need it. This is really important. So I want you to know that there are two different kinds of synthases. So let's briefly walk through the steps in nitric oxide synthesis. The endothelium are the brains of the artery. They sense when the artery needs to dilate or to contract. So there'll be agonists such as acetylcholine that are in the blood. Don't worry about it. Just know that there are things in the blood that the endothelium sense. Okay, they recognize these through G protein coupled receptors. So in our signal transduction lecture, I said that there are thousands of things that GPCRs do, and one of them is the regulation of blood pressure. So here's an example of that. So through GPCRs, in the endothelial cell, in an arginine-dependent mechanism, you'll generate nitric oxide. It's a gas. It can very easily diffuse to the smooth muscle cell. Once it's in the smooth muscle cell, nitric oxide activates guanylate cyclase. And I also mentioned that in our signal transduction lecture. Guanylate cyclase does lots of things, but it generates second messengers. So, let's summarize this. Endothelial cells sense circulatory factors in the blood. G protein coupled receptor, arginine dependent mechanism, makes a gas that diffuses into smooth muscle cells. It targets guanylate cyclase. That changes the calcium sensitivity of the smooth muscle cell, which we'll talk about in just about an hour. So this is the big picture, okay? Nitric oxide relaxes smooth muscle. So if any of you have grandparents that carry nitric oxide tabs, or my dad used to carry around it was almost like a stamp. He would keep it in his wallet, and if he felt some chest pain, if he had angina, put it under his tongue. By the time you count to 10, the pain will be gone because it vasodilates. It's very, very rapid, okay? And that's what you're doing. You're increasing nitric oxide. So what does nitric oxide do? It makes a smooth muscle cell relax, increases dilation to provide better perfusion of a particular muscle bed. And if it's your myocardium, it allows your heart to pump better. It does a lot of other things too. Inhibits leukocyte adhesion to the endothelium, so it makes it less thrombogenic. Same with platelets. It inhibits a lot of smooth muscle cell functions that we'll talk about in Block 8. So again, here's our pathway. There's our endothelial cell. In an arginine-dependent mechanism, you make NO. It rapidly diffuses to a smooth muscle cell, makes that smooth muscle cell relax. And if you can think of a cross-section of a vessel, if every smooth muscle cell is just relaxing even 10%, and you have thousands and thousands of smooth muscle cells making that vessel, you can imagine how the lumen of that blood vessel will relax. It's a really important mechanism. All right, been 15 minutes. Let's do a really quick review. Heterogeneous endothelial cells, you should know the names of the three types. What is nitric oxide? It's a gas. How is it made? What does it do? Main claim to fame, it's a vasodilator to increase blood flow. How does it do it? It targets guanylate cyclase in the smooth muscle cell. Why should we care? It regulates blood pressure. And if it's one of your grandparents with chest pain, it gets rid of the chest pain. Okay, so that's the first function. It's a secretory organ, and we focused on nitric oxide. Now we'll talk about thrombogenicity, leukocyte endothelial cell interaction. So this is how white blood cells will leave the circulation and enter the vessel, enter the artery, and then eventually enter you. It's also how platelets adhere. This whole process is called extravasation. 4 easy steps, and here's the cartoon, and I'll describe them as we go along. So you have a white blood cell, and they're circulating along their merry way. When it realizes a place where it needs to stop and leave the circulation and enter the artery, that's called rolling. It will stop zipping by, and it will loosely adhere, and it will roll along. Don't worry about the molecules involved. I want you to get the big picture down. When it realizes where to stop and extravasate and where to leave, it's called tight binding. That's the second step. So rolling is when it rolls along the endothelium. Tight binding is when it stops. The actual event of squeezing out of the circulation and into the artery or into you is called diapedesis, generally between endothelial cells. So you can sense right here when you have the discontinuous endothelium how it would make it easier for a white blood cell to leave the bone marrow, right? And then once it's in there, you have the last step is migration. So this is not empty space. That white blood cell then needs to get where it needs to go. It's not just going to roll there. It has to create a host of enzymes to dissolve away matrix in the basement membrane to be able to get from where it is to where it wants to be. So before I review, I think the most important part of this figure, and the most important part I think you should know, are these little green dots. Those little green circles are, I think, or what orchestrates the whole thing. They're chemokines, or inflammatory cytokines. So how, at what spot does this white blood cell know where to roll, and where to bind, and where to extravasate out? It's based on a chemokine gradient. So it's exiting the circulation here instead of up here because you have more chemokines, little green dots. And this is really important when we talk about atherosclerosis next September. 4 easy steps: rolling, tight binding, diapedesis, migration, all orchestrated by a chemokine or an inflammatory cytokine gradient. This is another EM. You can see a white blood cell squeezing its way between 2 different endothelial cells. But more than that, I have one of these YouTube videos. Now let's go like this. All right, hopefully you can hear it.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectins.
Don't worry about selectins.
Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly, and squeeze between endothelial cells without disrupting the vessel walls. Then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized mouse. The up and down movement of the frame is due to the mouse's breathing. 2 blood vessels are shown. The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins.
It's not true.
They do not crawl out of arteries.
She lies.
Some leukocytes are firmly attached and are in the process of crawling through the vessel walls, whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast under normal flow that we cannot see them. When the blood flow is restored, some of the leukocytes continue rolling, whereas all non-interacting cells are immediately washed away by the shear.
It's really pretty straightforward. Do we have any questions on that at all? Okay, so that's 2 of our 5 topics we want to cover about the endothelium. So the third one is maintenance of permeability. So the endothelium is an adaptable barrier. It's just not a layer of cells or saran wrap or a thin something that keeps things out or in. It needs to respond to its environment. In this situation, the environment is the circulation. So we can think of it as a semi-permeable, adaptable barrier. All right. So an important function is to regulate the transport of solids from the circulation into you, into the rest of us. And there are a couple different pathways. Don't get too hung up on this, but you need to know what they are. There's transcellular, where it's all based on size of the particle as well, too. Transcellular, where small molecules can squeeze between cells. It can also squeeze between pores, which we know are fenestrae, through an endothelial cell. Also, there's a pathway called transcytosis, where we talked about the caveolae. They're endowed with lots of receptors. That's a hub of signaling, but also receptors for other things like albumin and other proteins. So they can capture these, and they will bud off, almost the way a phagocytite can bud off and take something in. It's the same idea. Caveolae can bud off, form a vesicle, and transcytose its way to the other side. So the endothelium has to somehow know, and it's really cool how this happens. It has to know what to bring in, what to leave out, how it's going to bring it in. And again, here's our 3 examples: continuous, fenestrated, and discontinuous. So how this evolved over the eons is really cool. And again, think of the real estate rule. It does it, and how it does it, and what it brings in and leaves out is based on its location. So this is sort of a review of what I mentioned about 10 or 15 minutes ago. Fenestrae can be lined up in organs such as kidneys, and I showed you a really nice EM of that. They're called sieve plates. It allows you to very efficiently reabsorb water. It's the opposite in the blood-brain barrier. You really want to keep 99% of things out. So you want to form a really tight barrier. And it's the opposite in the liver and in the bone marrow. You really want to enable cells to get out of the bone marrow easily when necessary. So you can read this on your own. It's just a review of what I've said a couple times already. I said endothelial contraction regulates permeability. Well, we need to learn the mechanism. It's adaptable. So sometimes the endothelium will be more of a tight barrier than other times. How does it do that? And how does it know how to do that? It regulates its own cytoskeleton. So this is an important concept that I want you to know. Endothelial cells can pull away from each other by virtue of a complex called adherens junctions. Adherens junctions are located between endothelial cells. And the molecule on the endothelial cell that enables this, that makes up the adherence junctions, is called VE cadherin, vascular endothelial cadherin. So what am I talking about? You have a structure. This is the intracellular portion of the endothelial cell right there. That's extracellular. And there is a protein that traverses the membrane, kind of reminiscent of a receptor. And this is called VE cadherin. But instead of signal transduction when it's activated, it's connected to the cytoskeleton. So let's talk about this again. There's an endothelial cell. You have a structure called VE cadherin. It's built like a receptor. So you can imagine there'd be another endothelial cell over here with another VE cadherin. It doesn't signal, but it's connected to the actinomyosin cytoskeleton in that endothelial cell. So how does this work? You've got some resting conditions. So there's an endothelial cell right there. Here's an endothelial cell. Each one of these expresses VE cadherin on its surface. When they oppose each other, it's called an adherence junction. So think of it as Velcro right now. You've got two things stuck together. When you want that vessel, that capillary, whatever, to be more permeable, these have to pull apart from each other. So that Velcro needs to unzip. So what's the mechanism? Remember we talked about phosphorylation. We're going to talk a lot more about it in the smooth muscle cell lecture. But what phosphorylation does, it changes the structure, the function of a protein. So what's going to happen is there'll be a substance in the circulation that the endothelial cell senses. There'll be a receptor. That receptor will phosphorylate the cytoplasmic tail of that VE cadherin. It causes it to attach to the actinomyosin complex and detach from the opposing VE cadherin. And once it's attached to the actinomyosin complex, it will gradually retract away from each other. You now have space between those endothelial cells, and you can have more plasma that leaves the circulation and enters you. Let's go through that again. Opposing endothelial cells, they're zipped together. Adherence junctions are made up of VE cadherin. The cytoplasmic tail of the VE cadherin, upon appropriate stimuli, is phosphorylated. It attaches to the actinomyosin complex. It unzips from the opposing VE cadherin, and the cells pull away. A really good example of this would be like a bee sting. There are substances in a bee sting which are vasoactive. You get a localized edema, and you get swelling, and it's warm. Well, there are receptors on the endothelial cell that recognize, and here's my GPCR right there, that recognize what's in the bee venom, or thrombin, or histamine, or things that cause swelling. You have a signal transduction event that phosphorylates the tail. The endothelial cells will pull away from each other. You have edema. Any questions about that at all? You should know that. Now, I'm going to introduce another cell surface receptor that a lot of students get confused with VE cadherin. Don't get it confused with VE cadherin. We'll talk about it on the next slide. Another property of endothelial cells that are really unique to endothelial cells is called mechanotransduction. And when we talk about this, an endothelial cell has to have this. So they're always lining blood vessels. There's flow. The endothelial cell needs to know when there's disturbed flow. It needs to be able to sense that disturbed flow. So this is a property called mechanotransduction. And here's a nice picture. So you can read the heading on your own, but this is an example where you have blood flow going in one direction, and then in the aorta of that animal, it changes direction. And you can see the shape of that endothelial cell, rather rectangular right there. When the blood flow changes, the shape of those endothelial cells change to go along with it. No other cell type can do that. It is a mechanotransduction interface to respond to changes in frictional shear. I keep using the word adaptive. This allows the endothelium to be adaptive to changes in its environment. Well, here's another picture. These are endothelial cells in culture. You can plate them in a petri dish. Under static conditions, you have cobblestones. Okay, it forms like a tight layer. If you introduce a flow 24 hours later, they can reorganize their direction. It's a really important concept. It seems kind of mundane, but it's essential for an endothelial cell. We'll talk about why in just a minute, but now we'll talk about how. So I talked about VE cadherin a few minutes ago. Get in your head, VE cadherin regulates permeability. This guy is called an integrin. An integrin is a flow sensor. Integrins regulate mechanotransduction. Don't get them confused with VE cadherin, please. So here we are. We have this protein structure called an integrin. And think of it as sort of like a tree. So this is the ground. This is the roots of the tree. And when the wind blows, you have movement. It's mechanical, almost like signal transduction, right? But what it does, the inside is anchored to actin, similar to VE cadherin. But it's not going to pull the cells away from each other. It'll do other things. So these mechanoreceptors, these trees, these integrins, they're responding to the blood flow, allow the endothelial cells to sense shear stress. It's really exquisitely tightly regulated. It allows that endothelial cell to adapt to changes in flow. It can allow more flow. So think of physics. You've got the diameter of a circle. So think of this as an artery. And the smaller the artery, the more of a pressure flow you're going to have. You want to decrease the pressure. And there's a formula we all learned in geometry in 8th grade, which I can't think of right now. You increase the diameter, you're going to decrease the flow. It's going to need to know how to do that. So these proteins are called integrins. They connect the extracellular environment with inside the cell. It initiates signaling pathways, but it's based on movement. So when the outside moves, the inside's also going to move and initiate some signaling pathways. Why should we care? Why do medical students need to care? You're going to see the same exact slide in Block 8 when I talk about vascular pathophysiology, when we talk about athero. But this is huge. There are certain points in our vasculature, just how we evolved, that are more susceptible to disease, particularly athero, than other areas. And those areas are right at bifurcations. So we have a laminar flow here, which is all well and good. The path to bifurcation, we have disturbances in flow. They happen to be areas in our coronary circulation, in our cerebral circulation, and in other areas that we get plaque. And why is that? Because we have endothelial cells that are mechanotransducers. So in these areas where there are flow disturbances, we have distinctly different repertoire of gene expression. So where there is uniform laminar flow, we have atheroprotective gene expression. Don't get hung up on this now. There's plenty of time in September for you to understand, for you to know what they are. But where there's flow disturbances, we have expression of bad genes, pro-inflammatory genes, all made possible because of these integrins. It allows the endothelial cell, the endothelium, to be a mechanotransducer. So please, every year, medical students get integrins confused with VE cadherins and adherens junctions. This is for permeability. It's attached to the actinomyosin complex. Endothelial cells will pull away from each other. Think of swelling and bee stings. Integrins are trees sensing disturbance in the flow. You have gene expression. Think of athero, something like that. I know all the minutiae that you have to think about in this block and all the molecules that are thrown at you. Try to keep this in your brain. Sounds like an exam question, just saying, but what's the unique property of endothelial cells? Mechanotransduction, right? What are the mechanotransducer molecules? They're integrins. They sense changes in flow. They attach really the outside of the cell to the inside. Why should we care? A lot of vascular diseases are dictated by disturbances in flow. Okay? Lecture's going faster than I thought. I'm sure you don't care. We're going to talk about our last property. It's angiogenesis. If I'm talking fast, it's because I'm having trouble standing. Angiogenesis is we have new capillaries form by sprouting. Why should we care about that? It's really important. Wound healing, tumor metastasis, when you want to a myocardium, when you had an MI and you want to resupply blood flow, you want angiogenesis. So let's talk about this. Let's break it down. So we use the umbrella term angiogenesis, the formation of new blood vessels. There are different flavors of angiogenesis. We have physiological angiogenesis, wound healing. Okay, my knee has angiogenesis going on right now, hopefully. And embryogenesis. And the definition is of angiogenesis, sprouting from pre-existing vessels. So it's capillary growth. You have a layer of endothelial cells in this vessel, and we'll get into this in a few minutes. It decides it needs to put down a new capillary bed. They sprout. That's similar to, but different than vasculogenesis. Vasculogenesis is a type of angiogenesis, but it's not by sprouting. It's mobilization of endothelial precursor cells. These are constantly circulating less than 0.1%. But they will also sense a need for a new capillary bed. But you don't have sprouting, they'll get to where they need to go, and they will generate a new vascular bed. So the way I kind of describe this to another group of students is, there's your lawn, there's a bare patch. How's the grass going to grow in? If it grows in from the edges in, that's angiogenesis. It's sprouting of existing grass. You take a handful of grass seeds and you toss them in, and each one of those forms its own grass seed and its own little bunch of grass. That's vasculogenesis. The grass seed is an endothelial precursor cell. They're both physiological. It's a way to increase circulation. We have another form called pathological angiogenesis, and that comes into play with tumor growth. So we have a clump of neoplastic cells. A tumor is growing. And how tumors, one of their MOs is they will hijack the circulation to develop their own set of circulation. So these tumor cells will actually release angiogenic cytokines. And we'll talk about that in just a second. That will enable sprouting, this type of angiogenesis, to supply that tumor with blood. So let's talk about a situation. It's 2 years from now, you're across the street, you're doing your rotations. You're a cardiologist, and you have a patient who's had an MI, and they have an ischemic myocardium. You're going to want some angiogenesis, right? You're thinking of ways to really resupply that myocardium with blood flow. Or you're an endocrinologist, and you have a diabetic patient, and their circulation is really poor in their extremities. They might lose a toe or a couple of toes. You really want to think of ways to coax angiogenesis to resupply that ischemic tissue. A couple weeks later, you're up the street at Fox Chase, and you're doing an oncology rotation. You don't want angiogenesis. You want to find ways to stop angiogenesis. You want to starve out that tumor. So this is the thing that's going on daily. Pharmaceutical companies and scientists upstairs are finding ways to increase angiogenesis. Scientists and pharmaceutical companies are finding ways to stop angiogenesis. So when you're looking at your patient and you're thinking about a drug trial or something to enroll them in to increase circulation, you better hope they don't have anything underlying. So you should know about angiogenesis. So these are just a couple of examples of when you want angiogenesis. That's what I do. So here's a myocardium, and we'll talk about plaque and blockade, but it's blocked. What's the problem there? Everything distal from that blockage becomes ischemic. It's starved of oxygen and nutrients. So you have a little bit of a dead zone. You want angiogenesis, and one form of that is the formation of collateral arteries. So because these cells are ischemic, they send out signals, and you'll have the growth of collateral arteries to supply that area. That's one example of beneficial angiogenesis. Another one that also gets overlooked, and you'll see more and more in your practice as our population ages, is peripheral vascular disease. So it's really the same idea as what is commonly known as a heart attack. You have plaque that builds up, but instead of the myocardium, it's in a distal artery. It's in a peripheral vascular artery in the leg. And the patient will die of a heart attack, but when they walk, they'll feel pain. They'll feel crepitations. And it's really the same idea. And there's a really big push out there now to come up with strategies for neovascularization in the peripheral vasculature. It's sort of the same idea that I just showed you. I don't want you to memorize this or think of it, but you'll see it in your practices or when you do your rotations. Intermittent vascular claudication. It's pain in the legs. So if you have grandparents that complain of some pain when they walk, their skeletal muscles being starved, it's a little bit ischemic, and that's what they feel. Here is a really busy schematic. Just focus down here. So you have a muscle bed, skeletal muscle. You have a blood supply, and it's normal. And you have branches that supply that muscle. If you have an occlusion, you can see that that blood supply is shrunk, and the supply is also limited and reduced. This artery will respond to that, and you'll have angiogenesis. So if you just look up in this circle, you can compare the capillaries, the number of capillaries per unit compared to this capillaries per unit. And you're going to see a much more density, much more dense capillaries to supply that muscle and keep it going. So this is the whole point in trying to increase circulation. So if you have a diabetic patient, or if you have an older patient with plaque in the peripheral vasculature, this is what you're really after, what you're really trying to do. So we'll wrap up by talking about the cellular and molecular mechanisms of how this happens. Okay. So angiogenesis is controlled by factors released by the surrounding tissues. So the tissues that really need the increased circulation, they know that. I keep using the word they know that. This is how they know that. So when you have high oxygen tension, this is your tissue, everything's just fine. You've got a small blood vessel in proximity, and they stay there, it stays here, and everything's good. Under conditions of low oxygen tension, things happen. Now, if you recall in our signal transduction lectures, I talked about transcription factors. And transcription factors are related based on a theme. So I used NF-kappa B as an inflammatory master switch. We also have master switches for angiogenesis. A master switch for angiogenesis is a protein called hypoxia-inducible factor 1. Under conditions of low oxygen tension, HIF1 transcription factor is turned on. And some of the many things it does to enable angiogenesis is it makes chemoattractants. So when we talked about extravasation, the little green circles draw white blood cells out of the circulation. This is sort of analogous to that. You've got chemoattractants made by those cells that are slowly starving. Well, what does it attract? It attracts endothelial cells from a blood vessel in proximity, and they begin to sprout toward that ischemic area. So again, let's think about it. Under normal oxygen tension, everything is fine. When you have starvation of that tissue, of that muscle, whatever it is, a transcription factor called hypoxia-inducible factor 1 is turned on. Among some of the many genes it activates, it makes chemoattractants for endothelial cells, and they will sprout towards that starved area. So let's try to talk about these 5 steps for angiogenesis. We have hypoxia, growth factors are made. These endothelial cells now need to get there, from here to there. So they will make proteases to digest their way through the tissue to get where they need to go. So they migrate towards the source of starvation. They proliferate. You need to make more of them. They have to differentiate by forming tubes and these structures. And once those tubes and structures form, they can now bring blood to that area. And once you bring blood to that area, you return to normal oxygen tension, and angiogenesis stops. So this is just an example. I think over the course of 60 hours, they cause some tissue ischemia, and you can see all the sprouting that occurs from existing vessels. So it's a really tightly regulated process. You have low oxygen tension, transcription factor is turned on. That transcription factor makes chemoattractants. It makes growth factors. One of them is VEGF. Don't worry about that. The endothelial cells have to burrow their way from where they are to where they need to get to. They need to make more of themselves. Once they're there, they need to roll up and form tubes. Once they become functional, they deliver red blood cells. Oxygen tension rises. HIF-1 alpha turns off. The party's over. It's a really simple process. We're okay with that? Pretty straightforward. So this is just an example of some experimental angiogenesis. A lot of pharmaceutical companies do this. So they'll take an aortic ring from a mouse, a rat, a medical student, whatever. They'll put it in a dish, and they'll put in the growth factors, and then they sprout. So again, if you were an endocrinologist or a cardiologist, you want this sprouting. If you were an oncologist, you want to test different drugs to stop the sprouting. So we're done. Let's go through our objectives, and then I can answer any questions. So endothelial cells are polarized. They're heterogeneous. They have to be heterogeneous. You have so many different capillary beds and blood flows. A capillary endothelial cell is different from a coronary artery endothelial cell. 3 different flavors of endothelial cell based on location and based on structural features. Endothelium is a secretory organ. It makes things. We went into some depth about nitric oxide, how it's made and what it does. You should know that. Thrombogenicity and cell surface expression of molecules. We talked about extravasation. You should know the 4 steps of extravasation and what those little green circles are, because that's what I think is the most important. Permeability. V cadherin pulls away. You unzip 2 cells from each other. Plasma can escape. Mechanotransduction interface is mediated by integrins, and why should we care? Because that's where we get vascular disease, where there's disturbed flow. And finally, we talked about the different types of angiogenesis, physiological angiogenesis, pathological angiogenesis. You guys ought to know the steps in creating a new vascular bed. So I'm done at this point. I can answer questions. I'm not allowed to start the next lecture until 11. So sit tight for now. I'll be happy to answer questions.
smooth muscle tissue